Welcome back to this functional genomics course where we are discussing the topic what you call as comparative genomics, how comparing the genome helps us to understand how the genome functions. So, in the previous lecture we looked into how comparing the genomic sequence help us in identifying regions that possibly could function as a regulatory elements. It could be enhancer elements on which some protein comes and bind and regulate the gene transcription and so on. Therefore, if you have changes there, then the gene function may have been affected. So, we are going to look into one such example. Here it is a disorder caused in the human because of changes in the genomic region that are several megabases away from the gene, yet it affected the gene function and you have a condition. So, what is shown on the screen is a paper, and this paper talks about position effect due to chromosome breakpoints that map about 900 kb upstream and 1.3 megabase downstream of a gene what is called as SOX9 in 2 patients with campomelic dysplasia. So, this gene SOX9 is involved in 2 different pathways. One, of course, it is involved in the bone formation, cartilage formation. So, it is very important for the bone to form, but this gene also is involved in the formation and function of testis. So, in other words, in male it has got 2 functions for the testis formation and in the bone. In the female it has got only 1 function, that is for the bone formation. Now, if you have defects, then you would expect 2 things. One, if that individual is XY chromosomally and SOX9 gene is affected, then they will have 2 anomalies. One, they become XY female, meaning they could not develop testis, as a result they become phenotypically female. These are called as sex reversed females, and they will have condition wherein the bone deformities are seen. But if they are XX, the testis anyway will not develop, they remain female, but they will show what you call as the bone abnormalities. What is interesting is that a majority of the patients of course show mutation in the coding sequence of the gene. Therefore, the protein either it is not made or made in abnormal way, therefore the gene is not functional, as a result you have the defect. But there are patients in which, like that is discussed in this paper, where the translocation or structural changes happened about 1 megabase upstream or downstream of the gene, still it had affected gene function, resulting in identical condition like sex reversal and campomelic dysplasia. So, one of the hypotheses one can put forth is that these regions where the deletions or structural changes happened far away from the gene, yet you have the condition, could have affected certain elements that normally regulate the function of the gene. So, this and a couple of other papers I am going to discuss is to show how comparative genomics approach can be used to decipher the mechanism and test the possibility, right. So, this is one. So, what they have shown here is that these are the regions, this region and this region is where the breakpoint took place, chromosome got translocated, and it led to possibly the affecting the function of the SOX9 gene here. So, what they have, the hypothesis was that there could be a region here, right, which are elements onto which some transcription factor can come and bind and regulate the gene expression. If that is the case, if indeed that is the case, that these are the segments that represent certain transcription factors coming and binding, what you expect is that you have the promoter here, right, and then so you have the promoter here and then the elements are here and here. So, how these elements can communicate with the promoter that is present over here. So, what is believed and of course has been shown in other gene segment is that this is your promoter, and it possibly this DNA can loop out and bring the elements, for example, the upstream element and the downstream element, something like this, and you have the complex of proteins coming and binding and regulating the gene expression. This is one of the hypotheses. If indeed it happens, then if you look for the distance between this and this segment in a cell that is normally expected to function and the SOX9 being expressed, then the distance should be much less as compared to a similar region in some other gene where there are no elements like this, right. So, that is what tested in this particular study. What they have done is that they have looked at, for example, a gene. They have taken a gene which do not seem to have any such control elements, and then they looked at the distance between the gene and probes that are falling about 1 megabase away from the gene, right. And that is what is shown here. You can see here in this blue circle is nothing but a nucleus, right, of a cell, in which they have used a red color probe for the gene and a green color probe for a region of the chromosome about 1 megabase either upstream or downstream. Okay? And you can see the distance between the green and red is far apart. These are two copies that you can see here. And then they are looked at another gene, which gene is known to have such regulatory element, meaning it has a regulatory element about 1 megabase upstream of it, and they looked at the distance. You can see that they are almost present close to each other. That is because of this particular model that I am proposing, that they come together as a complex, the protein bind and activate the gene. So, the distance is almost negligible, although they are separated. The distance between these two segments is about 1 megabase, but they loop out to form a structure wherein these two elements come together. Now, they tested whether it is same is true for this case where SOX9 is either the breakpoint here and here affecting certain element, and that is what is shown here. For example, here in both these conditions, right, whether you are using a probe for a downstream of the SOX9 gene or upstream of the SOX9 gene, you will find the distance between the two signal, that is red and green, is almost similar to what has been shown for another gene which is established to have a control element about 1 megabase outstream, right. So, it clearly tells that there could be certain elements, functional elements present far away from the gene, and if that is disrupted, the gene will not function, right. This is the paper that proposes that a possibility. Can you use functional genomics or approach to test this possibility? This is one of the earliest paper where they have looked at such possibilities, and what they have done is first they compared the sequence of the human with the mouse. Okay, this is the SOX9 gene, exon 1, 2, 3, and possibly this is where the upstream regions are there, and you can see that this is 50 to 100% similarity, and you can see that there are segments, right, that are highly conserved. You can see that this is compared with human and mouse sequence compared with puffer fish. Okay, a fish which has got again SOX9 gene and separated by millions of years from the mammalian species, and they compared the pufferfish genome with mouse and human genome, and they are showing that exactly at the same about 18 megabase or what a kilobases, and you have segments that are highly conserved, right. So, which suggests that these are the regions possibly there are elements that regulate the SOX9 gene expression. They are conserved because change is possibly not permitted, otherwise the gene activity may be lost. Okay. Now, this is simply to show that there are comparisons like percent identity between, for example, the pufferfish and the mouse, like what I have shown earlier. But now this paper which came out more recently is they tested that whatever has shown conservation, meaning higher sequence similarity suggesting that these are control elements, do they really regulate the gene expression in the human? So, how will you test that? So, they have created what you call a transgenic models. Okay? You take the promoter and other control elements of the SOX9 gene like shown here. SOX9 gene you have elements here, 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 here and here, here, right. There are 7 different elements that are known, highly conserved, likely regulate the gene expression. What you do is that you take these elements, make transgenics. In each one what you do, you delete one of the elements and see what happens to the expression of the gene. So, now this is what that has been done. So, you have this SOX9 promoter tagged to a coding sequence called LAGZ, which is exactly like what you guys use in bacterial blue-white color selection. So it is an enzyme beta galactosidase, you give a substrate, it will give you blue color. So this is a way to assay where the gene is expressed. You don't need to really worry about SOX9 here, because you are using the promoter, which if you get a laxy blue staining, that is a readout for the promoter's function. So this is what it is. Now if you use just the promoter, okay, this region alone with the laxy, and if you look into where the laxy is expressed, you can see that it is the hindbrain and spinal cord. This is what I am showing here. This is the embryo. The embryo you can put in a substrate having the beta gal, it works on that and converts into a product which gives you a color, and you can score now where it is expressed. But if you take SOX9 promoter with E1, like what is shown here, if you make a construct, and you find that it is in addition to the hindbrain, it is also expressed in other tissues like notochord, gut and others. Like you can go on seeing like this. As you add, for example, E2, then you do not see a difference, but E3, E4, E5, you find that it is started expressing in cranial neural crest, inner ear and other. So you can see that they are, how in development, the different elements control the expression of this gene in different segment. And what you are doing is, you have taken the genome of the human and you are putting it in the mouse. Still it is able to drive the message as to where it should express. So it is the power of the comparative genomics. You go and compare the genome, you identify the signatures, predict it could be elements, and create transgenic models to test your hypothesis. So this is all doable only if you are able to compare the sequence, and that tells you how powerful the comparative genomic approach is. So let us look into the genome. So if you compare the different genome, what is that one thing that strikes you the most? With regard to the human genome, what you find is that the gene density goes down. So this is a bacterium, for example, a chromosomal segment of 20 kb of bacterium, yeast, Drosophila and human compared, and you will find that for the same segment you are going to have a large number of genes in other species, whereas in humans, so for example, you have only 3 genes, but here these are multi-exonic, so it is spread out, right? Whereas in others you will find these are much more clustered and packed, okay? In other words, the number of genes did not change really dramatically across species, but it is this the other DNA, the so-called non-coding DNA that is there in the genome that is anomalously increased, therefore that possibly have got some strong control over how the genome functions. So one of the important components of your genome is the transposable elements, or there are what you call as jumping genes. There are segments in your genome which are called as transposons, because they are more like parasites, in the sense they are not there to help our genome, but they are there for their own survival, because they have to survive, right, because they are parasites. They do not do any harm to us often, but at times they can harm us, at times they can help us. Both happens without any direction. I will say why it is so. These are transposons, nothing but they have a small segment of the DNA which has a coding potential for a reverse transcriptase or other nuclease. What it does is that it makes copy of itself and goes and get integrated somewhere in the genome. So they keep on moving within the genome, leaving a copy. Therefore, over the time what would happen, you have accumulated a large amount of such sequence in your genome. It's increased, genome size has increased, and and they are all in between genes. The reason is, if they have landing in part of a gene, then it is going to affect the gene function, and that may affect the survival of the individual, therefore it is not selected. But wherever they are integrated in a region that are not having any gene, your genome tolerates. So that is what is shown here. So if you look into the protein coding sequence between human to yeast and others, it is not a big difference, almost similar. And then if you look into the genomics size, the entire DNA, and obviously in the human mouse and zebrafish, you can see that is like a tower, because the genome size is very large, and so is the case with plants. They have as big as the human genome size. And in addition, what you have is that a good proportion of almost 50% of the genome is made up of these kind of transposable elements. In other words, the transposable elements contributed to the genome diversity, and also the increased genome size. And there are elements that are unique to species. Some of the transposable elements that we have are unique to humans. For example, ALU transcript, the repeats that we have. these are unique to human, you don't have it in other species and so on, right. So that really changed. So as I told you, the transposable elements can seed some evolution, okay. So evolution could be, at times it can land, as I told you, in a gene and affecting the gene function that can result in a disease or the individual not surviving, but it can also help the genome to survive. One of the ways by which it can help the genome to evolve is by facilitating a process what is called as gene duplication, okay. So what happens in this case is that you could have, for example, the element landing on either side of a gene, right. You have identical sequence present on either side of the gene it happened. So what happens during meiosis, it could so happen that there is a process called recombination that happens in recombination. So in the meiosis, and the recombination normally looks for identical DNA sequence and then it brings the homologs together. There is a double strand break, therefore there is a crossover happens. So since the elements, the transposed elements have identical DNA sequence, at times what happens is that you could, if you have two such elements nearby, the system may get fooled because it may identify these two as homologous segment, and then you would have a recombination going through this. As a result, one of the two recombinant chromosome would have two copies of the gene, other would have lost the gene, right. So now when you have two copies of the gene, you have the luxury. The luxury is that instead of having one copy in a chromosome, you have two. So one is doing the function of original function, the other one is free to evolve because it changes, is not compromising the gene function because the other one is taking care. So now if the changes can result in such a way that the gene becomes dead, meaning accumulated mutation, no longer functional, or it can give a new function to the genome or the gene, therefore it evolves into a new gene, right. So this is a possibility. That is what is shown here, is that at the end of the day you could have, for example, there is a protein change, this sequence has changed dramatically, that this has become almost a different protein, or the expression changes where the gene is expressed, that could change, or it can completely lose its function. So what I am showing you is that how such elements help in the genome. Now there are large number of transposable elements in your human genome. Some are shown here, lines, signs and other transposons, but what I am showing you is a classic example which you must have studied in your textbook. This is a diagram schematic which compares the yeast genome with the human genome, right. So the human genome that is compared here is from chromosome 11 having the beta globin gene cluster, because this is one of the gene dense region. Even then you can see the number of boxes which represent the gene are fewer in case of the human. Obviously you have more spacer elements. These arrows are nothing but the repeat elements. Remember I told you that there is a transposon called ALU, repeats, and these are repeats now that are present here. So what we believe is that these ALU repeats helped in the evolution of beta globin genes. Multiple forms of the beta globin that you see could have happened because of gene duplication, each one now selected into new function, and some of them would have lost the function like what you see here. So that is one way, and the same kind of hypothesis is given for hemoglobin as well. So you have the adult hemoglobin and fetal hemoglobin. The difference between these two is the fetal hemoglobin, the hemoglobin that expressed when we are embryo growing inside our mother, is a hemoglobin that has a higher affinity for oxygen, right? Therefore it is able to capture the oxygen from the mother, whereas the adult hemoglobin has lower affinity, because now you are able to capture oxygen from the atmosphere. So this also people believe could have evolved because of gene duplication, and the duplicated gene acquired something new function, right. So how do you really study, right? When I talk about transposons, right, transposable elements, one of the model system or one of the species that has got very few transposable element is the pufferfish. That is something that is shown here. You can see that this fish called fugu, has got negligible, amount of transposable element, and the genome is so small. It is just 400 megabase, 1/10 of the human genome, but it has got as many genes that human has, almost all number of genes, right? And then the size of the gene is 6 to 7 kb, pretty small. In other ways, it is like a miniature genome, which has got all the genes packed in a very small segment. So that has really made people to wonder what is so unique about this fish, why it has got such a small genome. And if you can sequence the genome, you can understand what are the essential genes, what are the essential DNA elements that are required for a successful species, right. So that led to this whole genome sequence of this fish, Fugu rubripes, and this is a fish that is normally found in Japan, and this is one of the delicacies. People eat this fish, and this was developed by the person Sidney Brenner, who received Nobel Prize for his work on C. elegans. Okay. So as I told you, this fish has got very few transposable elements in the genome, and probably that could be one of the reasons why it is able to keep the gene density very high. Okay. So what has been done is there are many studies one can look into. I have given you another example how this pufferfish helped in understanding SOX9 control elements. So what we are going to discuss today in this section is to see how the fish genome can be used to understand what possibly was the first vertebrate genome. So when vertebrates come in, the first species of fish evolved, what could have been the chromosome, how many chromosomes, how it could have been. So what people do is they make synteny maps, meaning you look into the chromosomes of human and look into the chromosome of other species and see which segment of human chromosome represented in which region of the chromosome of the species you are studying, because the chromosome number is not same. So we have 23 pairs, but there are some species which got only 6 pairs, mammals, same amount of DNA. Some has got 100 plus chromosome, which clearly tells there is the genome has undergone large number of fission and fusion process, meaning if chromosome cut into multiple pieces and joined independently form new chromosome, that is the fusion process, right. So you can now look into the genomes where the genes are and the clusters, and then go and map in the genome. So this is what's shown here. For example, that the fish genome was mapped to the human genome. So this the human chromosome is color coded here, and for example chromosome 1 is mapped to you can see that where they are present. It's present somewhere here, present somewhere here. There are large number of segments in which chromosome 1 is present. Likewise, every other segment. One can do the reverse. You can take the chromosomes of the tetrahedron, the fish chromosome, 21 pairs, and then look at which are the segment that in which it is present, right. So this is the way you can analyze. And when you compare a fish with a human, it looks like scattered, right. But if you compare a mouse with human, you will find there are many regions that are present in a larger segment like this. And based on that kind of a comparison, what has been done is that this is the human chromosome. They went on to sort of propose a set of what is called as ancestral karyotype, chromosome set for the bony vertebrate, because that one, the bony vertebrates are the one that are evolved for, from the soft-bodied fishes, and that led to massive genome duplication, leading to the kind of fishes that you see. Okay, so this is the ancestral karyotype. They believe that there could be 12 chromosomes, right, which is numbered from A to L, and what had happened was that the genome, obviously this genome was much smaller as compared to human, but what could have possibly happened is there is an amplification of the genome, not because of duplication, the whole genome duplication, but because of transposable elements. They came into the genome, kept on leaving a copy and making a new entrance, so on. So the translocation and the genome size is increased, and then what had happened is that you have chromosome fused together or broke with each other, resulting in the kind of chromosome that you see. So this is an hypothesis based on such kind of analysis. But what is interesting here is in the fish, the genome evolved in different way. What happens is the entire genome got duplicated. So for a given gene, now it has become 2 in number, and then again you have process which led to the fission, chromosome broke, and join together and to see what kind of chromosomes that you have. So we can really understand how the genome has evolved by comparing the genome sequence. That is what we spoke about, how for example the pufferfish helped us to understand the evolution of the genome with little amount of what do you call as the transposable element, right. So, by tracing the evolution we can now look at some of the major events that happened. One of them is that the in the mammals the evolution of X and Y chromosome. So, if you when you talk about sex determination, you have X chromosome and Y chromosome that determine the sex of the individual. If it is XX it is a female, XY it is a male. It is common to all the true mammals, right, including for example the kangaroos, right. So, what we believe is that that happened sometime at this point of the evolution, that a given chromosome was identified to become the future sex chromosome, and they were identical in size, but over the time point when the evolution progressed, it became such that the Y has become very small, X has become very large, and Y invariably had the gene that determines the testis. So, now we have very good leads which suggest that indeed that could be the case. The other important element that people have dissected is domestication. For example, the pigs were domesticated. There are very good number of studies which tell you how the genome has changed while domesticating the pigs, because you have selected for certain quality in the domesticated pig, because you are looking at certain quality, how the genome has changed then. And then of course the language adaptation, right, in the humans. So, for these two I will give you some links in the course portal. You can read more on that. We will not discuss more, but we are going to slightly touch upon the language adaptation in the human, how your genome probably helped us to develop the language that you speak now. So, we know that there are individuals in the human population who are unable to speak the way majority of us speaks. So, that's called as speech or language disorder. They are unable to comprehend, understand the language, or unable to put together words to express themselves, right. We are talking about not a non-native language, we are talking about the mother tongue language, we are not talking about which language it is. So, and looking at such families, there was a study which identified a gene, a defect in that gene, led to such kind of disorder. Obviously, genes regulate your cognitive abilities, one of them being the language, right. So, if that gives you a key as to how genes can regulate your brain, right. So, if that gene is so critical for language, then you can go and look at the gene sequence and ask whether the sequence that you see in the human that made us to speak the language, develop the language, understand the language, these signatures are present in the other animals which do not use the kind of language that we use. That gives us a glimpse as to how it could have evolved. So, now what they have done is a similar study was done. So, there is a paper in 2002 where they looked at this gene FOXP2 gene which is involved in this severe speech and language disorder. They looked at the evolution of the FOXP2 gene across different species, and I am just putting this paragraph here, you can read that. So, what it says is language is a uniquely human trait, because the kind of language that we have no other animal has, likely to have been a prerequisite for the development of human culture, because you have what do you call as inheritance. You are reading about somebody, Mahatma Gandhi or Shivaji and so on. We are able to read because there is a language. So, nobody need to tell us, we can open a book and read and understand what it is about. So, you can leave the history because of the language, otherwise you would have forgotten. It would have been just like our epic, like Ramayana, Mahabharata, which was told by the parents to the kids, that's our remembered, but that's now the information has grown. Now, everything cannot be told verbally, so you need to have a language. Even for verbally telling, there is a language, right. So, everywhere there is a language. So, the ability to develop articulate speech relies on capabilities, not only the cognitive functions, such as fine control of the larynx and the mouth. Basically, that sound that you create is nothing but the sound you are able to control and produce using your mouth, because you are closing, your tongue because you are twisting the tongue, and you are able to use the larynx because that is where you make the noise, or the signal, which is what you call as the language. This ability is absent in chimpanzee and other great apes. Okay? So, the FOXP2 is the first gene relevant to the human ability to develop language, because you have a defect there, you are unable to speak. A point mutation in FOXP2 co-segregated with a disorder in a family in which half of the members have severe articulation difficulties accompanied by linguistic and grammatical impairment. They cannot speak and they cannot comprehend or put together even a sentence. This gene is disrupted by translocation in an unrelated individual who has a similar disorder. So, in more than one family, the same gene defect resulted in a phenotype, which is you are unable to speak. So, the functional copies of the FOX seem to be required, that is, you have to have 2 copies required for acquisition of normal spoken language. It is very important. So, what they have done is they have sequenced the complementary DNA that code for this gene FOXP2 protein in chimpanzee, gorilla, orangutan and other monkeys, apes and others, which do not have the ability like us to articulate, okay, and compared with the human cDNA and to see what happens. What they found is that we also investigated the interspecific variation of the human FOXP2 genes, and what they have shown is that the FOXP2 contains changes in amino acids coding and a pattern of nucleotide polymorphism, which strongly suggests that this gene has been the target of selection during recent human evolution. That means the ability that we got to speak is because of this gene, and some changes happened, and the changes could be not really meant for this gift that we are able to speak, but it so happened that that gave us this ability and we picked up and this was selected. Now you find it in humans, right. This is just to show you that signatures, you can see that there are variants, right, and these are the variants that are unique. You can see that this S amino acid is unique to human, and likewise here is an amino acid is unique to human, and there are segments that are multiple Qs. People believe that these are the residues that has given you that ability to speak, okay, whereas the other animals do not have the ability. Possibly that is the signal that led to the language. It could be very subtle small change, but it can have a greater impact. Another example is that our brain itself. So what you can see here is that although the orangutan or apes have much larger head, but their skull has got small cavity for holding brain. So if you look into the skull volume versus relative brain size, we have got the larger brain size in the skull, right. So what led to this evolution, and we don't know for sure, but what we believe is that it is the ability to use your hands as a tool could possibly one gift. So what is seen here is that skull of a human and skull of one of the apes, and you can see here that although the head is so big, there is a huge cavity there, and it is there because the lower jaw of the apes are so strong, right, because they have the muscle connecting from here to here, and for the muscle to connect you need a huge cavity there. So if you don't have the cavity, the muscles cannot connect the lower jaw. As a result, what happens, the lower jaw becomes less powerful, so they can cut open anything with their lower jaw. And possibly there was a mutation that happened with the loss of the muscle strength, which led to a condition wherein you don't really need that huge pocket. Therefore, the skull now is allowed a larger brain to occupy. Now it comes with a cost. The cost is, so you do have a bigger brain the same size of skull, but your lower jaw is not as powerful as it was. So now what you need to do? So you need to use your larger brain to compensate the loss of the power, that is to make tools. What you can cut with your mouth, you can cut with a tool. So that larger brain helps you to come up with tools. So you compensated, but it really helps you to weigh the way the humans have evolved. So it's an hypothesis based on the sequence analysis. What is shown here is a sequence analysis. You can see there are again unique residues that are present in case of humans but not in other species of a gene that codes for the myosin, one of the protein that help the muscle, and people think that that could possibly be the one of the reasons why your skull is able to accommodate much larger brain. Okay, so that's about how the humans evolved. Now I'm going to talk about some unique species, the species that have power to live beyond what the human can imagine. So I'm going to talk about one ugly animal, looks ugly, but the phenomenal animal in terms of its ability. This animal that is shown here is called as the naked mole rat bat, because it doesn't have, the skin is open, it doesn't have these hair, so it's naked. And it's called as a mole rat because they live in burrows, and mostly in dark conditions. They don't have the kind of eyes that we have. They can sense light, but they cannot really see beautifully. But they are amazing animals. They live in burrows like this. You can see that all these are small litters that these are mammals, just like the size of mouse or rat, and they are unique to this eastern part of Africa. Okay, so what is so great about it? Therefore, people went and sequenced the entire genome of the naked mole rat. I'm just putting you one paragraph again to appreciate what is so unique about this naked mole rat. Okay, it's strictly subterranean, meaning it lives on the surface of the earth. Extraordinarily long-lived mammals. Although the size of the mouse, its maximum lifespan exceeds 30 years, making this animal the longest living rodent. Naked mole rats show negligible senescence. They don't age at all. They look perfectly normal. No age-related increase in mortality. It's not they die more at later part of their growth. High fecundity because they can keep on breeding until death. So, they are reproductively active until they die. So, they don't age. They live longer. They are absolutely all right even at the time of death otherwise. In addition to delayed aging, they are resistant to both spontaneous cancer and experimentally induced tumorigenesis. So, the genome has got something which make them resistant to cancer. So, they are able to somehow control the bad genes from going on and inducing cancer, even if you experimentally induce. These rats pose a challenge to theories like aging, because we normally say aging is inbuilt in your genome. You age because the system wants you to die. You have done your job of reproduction. You no longer compete with your offsprings. You have done your duty. That is the concept, but it defies that even theories. And then cancer, they say that it happens because the death has to happen, and redox homeostasis because even your ability to cope up with the stress, cellular stress, goes down with aging, but nothing happens in this animal. Although characterized by significant oxidative stress, the naked mole rat proteome does not show age-related susceptibility to oxidative damage. No damage happens. There is no increased ubiquitination saying that proteins are damaged. Nothing happens here. Naked mole rats naturally reside in large colonies with a single breeding female, the queen, very similar to what you see in honeybees, who suppresses the sexual maturity of her subordinate. Therefore, she remains the main female who is reproducing. They also live in full darkness at low oxygen and high carbon dioxide concentration because they live in furrows, and are unable to sustain thermogenesis nor feel certain types of pain. It all gives, because when you feel cold, you shiver. The shivering is a physiological response to increased body temperature. They don't show such kind of shivering when you bring down the temperature, and they don't feel pain either. So, it is a gifted organism which has sort of whatever we think of they should get rid of, they could get rid of, what is there in the genome. So, what they have done is they have sequenced the genome and analyzed the genome of the mole rat, which reveals unique genome features and molecular adaptations consistent with cancer resistance, poikilothermy, you can survive in different temperature regime, hairlessness, you have fur for protection, they don't need that. Insensitivity to low oxygen, they live in hypoxic condition, very little oxygen, they are survive better. And altered visual function, which you cannot see but still they function. Circadian rhythms and taste sensing all are compromised because you don't have the daylight, so the circadian rhythm, what you normally see in mammals, all altered here. So this information provides insight into the naked mole rate's exceptional longevity, ability to live in hostile condition in the dark and at oxygen, low oxygen level, right. So we can understand how the genome functions, what do they really mean, how this animal becomes so successful, possibly it could help us in advancing our own biomedical research. So this is just one Venn diagram I am showing to show that how this particular species has got genes that are so unique, very different from mouse or human or rat, but unique to this species. If you can understand the function of these genes that are unique to this mole rat and what function they do in the cell system, tissue system at the organism level, probably you will get some understanding as to how we can control cancer, how we can live in conditions that are hypoxic, and many other advancements that otherwise of very challenge to the human being. So this is what being investigated. These are some of the flow chart people are trying to understand, for example, long lived and short lived. Animals compare the transcriptome, genome, decipher what is the difference and go ahead and understand the function of the gene in the physiology. That is just to summarize some unique species, how they can give you some insight into some of the physiological conditions that otherwise are extremely lethal to the human. There are similar approach have been applied to plants as well. So you can, for example, you can look into species, right, A and species B, some are large or small, and compare the genome and understand what genomic changes gives you particular trait. For example, you talk about cabbage, right, it's a flower like thing that we eat. If they can make much larger, some variety makes smaller. So if you can understand what is the genomic signature that gives you the larger cabbage, possibly you can engineer plants. Or the leaves, for example, spinach leaves again. So a variety that makes huge leaves, if there are signature you identify, you can get it. Likewise for tea plant, coffee plant, you can go on for flower and so on. So this is another line of investigation. One can study plants for the unique phenotype that could be of advantage to the humans, and also the parasites, for example. The parasites have evolved from free living species, right. So you have species that are parasites, there are closely related species that are free living, they do not infect any organism. So if you can compare the genome of a parasitic species with a non-parasitic closely living species and compare the genome, you can understand as to what are the genes that has given that ability to become parasite. So then you can target those genes or the gene products to control the parasitic infection, right. So this is another advantage directly to the human that you do. So the approaches what we discussed are just gives you an idea as to how one can approach. The outcome is endless. It all depends on what you are looking at, how you want to do, what are the technologies developed. So it is unimaginable. In next 10 to 20 years you are going to see a revolution in functional genomics, because this is just the beginning where we are trying to understand the genome at large, how it functions. It regulates the gene expression, protein expression, how the cell functions, tissue functions, and organism functions. And if you can understand likewise for a variety of species, possibly you will get more insight and help us in either preventing disease or controlling the disease, or increasing the desired phenotype in the plants and animals that we are directly dependent on. So you are here to witness such changes in next 10 years. It's going to be very dramatic, and if you want to look into any field that's going to really make a huge impact on the human society, it is the functional genomics. And thanks for joining us in this course. I hope you enjoyed.